Okay, Pinaki, I'll get started. So good morning, everyone. Really thank you, everyone, for making the time to hear this recruitment lecture. This morning, we have the honor of having Dr. Pinaki Sarder, who's an associate professor in the Department of Pathology and Anatomical Science at the University at Buffalo, the State University of New York. His background is of an electrical engineer. He became an electrical engineer at the Indian Institute of Technology in Kanpur, India, and actually obtained his PhD in electrical engineer at WashU in St. Louis. While his time in St. Louis, he trained in many areas related to radiology, including optical radiology. He was also a research fellow in the Department of Biostat at Harvard. And he actually did some training in imaging science while at WashU, and joined the Department of Path and Anatomic Science there, and has gone up the rank, and currently is an associate professor with tenure at the University at Buffalo. He has received numerous awards and honors. Among those, he's the recipient of the University at Buffalo Exceptional Scholar and actually Young Investigator Award. He has been invited to many national and international meetings, including NIDK seminars on computational image analysis in nephropathology. He has mentored a good number of PhD students and master students. His current funding is all related to kidney disease, including funding from the Kidney Precision Medicine Project. As we all know, this is a major NIH initiative to look and interrogate kidney tissue. He has also acquired funding from the CKD Biomarker Consortium on lupus nephritis. And he also holds an R01 on imaging of renal structures for diagnosis of diabetic kidney disease. He's very well published. He's very much interested in machine learning in the areas of diabetic kidney disease, GNs, as well as lots of interest in kidney transplantation. So Pinaki, thank you so much for making the time. After this talk, Dr. Sarder will have some interviews with many members of the AI department, Biostat department, as well as the renal division. So thanks again, Pinaki, for making the time, and you can proceed with the presentation.
Thank you, Dr. Chanchal, for the introduction. So I will discuss today the computational pathology works in our lab at University at Buffalo toward understanding better chronic kidney diseases using AI tool. So before I start, let me tell you a little bit about the University at Buffalo. So we are located at this northeast side. You can see my pointer, right?
Yes, very well.
So we are located at this northeast side of the United States, and you possibly know of Buffalo because of the Niagara Falls. It's very nice here in the summer, and I hope I mean, there are places also other part of the US as well. I'm pretty sure Colorado is as nice as Buffalo. So we have three large campuses. North campus mostly host these engineering departments. Then you also have a south campus that is in the middle of the north campus and the downtown campus. It hosts the School of Public Health and Dental School. And then I work from here. This is a new facilities in downtown Buffalo. It's a brand new facilities for the medical schools with many state-of-the-art and cutting-edge facilities. Today I'm not speaking from here. I'm speaking from home, but that's what the location is. So my interest is in the detection and estimation of features with biological relevance from medical images. So an image is composed of thousands of pixels measuring texture of objects that can eventually be summarized using statistical measures for quantifying the underlying information content. So let's pretend that we have access to that system which can break the fundamental resolution barrier and can see us infinitesimally small objects. And question then would arise how to associate information across scale, and that is the fundamental problem in any biomedical problem, to fuse information or integrate information across the scale. And this thing, I mean, is very nicely explained using this famous painting from the pointillism era by Surat. So, but we are in a revolution today, because we have photonics revolution going on. It's almost started like in 2010. Then omics has reached to a place where it's a technology, and then we have modern advent of this AI because of this these GPU technologies. And the data, the amount of data we have available is huge, and all the NIH consortiums like KPMP, HubMap, they're generating huge amount of data. So the question is that how we can use this data and decipher the underlying molecular information based on the information available in multimodal medical images. So that makes me start my, that initiated my interest in this area, and I've been working on these dimensions or these directions throughout my life, slowly by slowly acquiring knowledge from one domain to another to get to that level. So when I started working on this as an independent faculty at University at Buffalo, I had to focus on a specific disease area, because we had to get tenure, and then funding is mostly NIH funding is mostly focused on the disease. And I was very glad that I started working on kidney. It's a great organ to work with. During my postdoc, quite unknowingly, when I was doing some imaging, I used to choose kidney because of the interesting structures inherent there, which are very well patterned. But then during my PhD, during my independent faculty position, I started working on this thing called diabetic nephropathy. So it's a disease secondary to diabetes, and we at some point it gets to end stage renal disease, because kidney gets deteriorated, and it has caused a huge amount of healthcare burden. Accounts for more than 19,000 in Medicare costs yearly for each patient. And this data is coming from few years before. I don't know the current numbers. But the problem with DN is that it manifests with damages to the renal glomerular structures and other structures that often may not correlate with clinical biometrics. So to give them more detail, these are the Tarvert classification. If you are in kidney domain, then you would possibly know the Tarvert classification scheme. It's a classical classification scheme developed by Renal Pathology Society, which shows the structural change in renal glomerulus, right, from in different stages and the structural pattern. So stage 1 doesn't show too much of change in light microscopy, but in stage 2A you see small changes in the mesengium, then more and more changes in the mesengium. You get nodules here, and in stage 4 it completely does not function. But the problem here is, that some people with stage 3 may live as long as they're possible, but some people with stage 2B may progress to the end stage. And it also has to do many things with age, sex, ethnicity. And I'm not going to talk about at that how we can see these variations with different diversity in the data with respect to age, sex, ethnicity, but that's also important. And the question that I started asking is, what are the earliest measurable image features in DN renal biopsies that can predict progression? So that being said, in these directions and subsequently very other chronic kidney disease research, I recently started with a little bit of AKI because of the KPMP involvement. So we work on very large tissue image data, and we develop computational image analysis tool for detection, segmentation, and feature extraction of microanatomical structures from this large disease tissue image. So this is a kidney biopsy, possibly you know already, and a small structure is glomerular, and this image can be large. So this is digital image data, and we work with this large pixel data, and we have often learned something about detection, segmentation of structures. So we are currently going beyond segmentation a bit, and we are trying to study computational correlation, integration of computationally extracted image features with diverse other data types, including outcome and molecular data. And we have done extensive work with understanding outcome as well as correlation with urinary proteomics data. we have we work with other data type as well. For example, in addition to EM, single cell or single nuclear RNA-seq data, spatial transcriptomics data, maybe metabolomics data as well. So I don't know how possibly most of the folks here from the nephrology, but if there are any engineers present here, I mean, I always go to the fundamental. So we humans have, so let's look at this basic renal physiology. So we humans have two kidneys, and these two kidneys are used for renal filtration, as we know from high school textbook. And every kidney is every kidney has million of this kind of unit, which is called nephron, and it has a bowl-like structures called glomerulus, where blood vessels come and form entangled loop for filtration, and then the vessel goes out of the body. The next part is proximal convoluted tubule, loop of Henle, and then this distal tubule, and the blood vessel also has exchange in this tubular region before it goes back to the body. And any exchange liquid along with waste go out of the body via the collection duct. So in detail, the flow through glomerular barrier is shown in detail here, where when things are damaged, these are large and small proteins, then sometimes small proteins pass through that in the tubular region, but small, they are mostly deabsorbed. But when there are damage here, right, so these proteins that get they get leaked in the urine, and that is called proteinuria, and that is one of the symptoms of of somebody's kidney is not functioning. So again, for students who are from engineering, I mean, I like to go to basics. So we show the schematic of renal glomerular cross section in 2D. There is one thing wrong here. I mean, I never fix this, but I will talk about that. So I think so this 2D schematic here shows glomerular cross section with different cell types. The capillaries are shown here with this endothelial cell. There should be one endothelial cell. I showed multiple, and then you can see there are RBCs here. So this is where the blood flows. This is this 2D cut of this 3D structure. Then there are small pores here through which the exchange happens. The overall capillary is marked by the capillary wall, which is surrounded. So this is the capillary wall, and there is a cell called podocyte, which is a very important cell where extraction and contraction happen to help release the things in the urinary space, and this whole thing is surrounded by another cell type called parietal cell and Bowman's capsule, and this whole thing is arranged in 3D using something called mesengium. Okay, so and another thing I enjoyed very much when I started working on the topic, to see in pathology, hand in hand correlation of these structures to make a thorough understanding of the structure. So this is a glomerulus, and you can see that this is a mesengium cell because this is inside this mesengium. This is a parietal cell. This is your urinary space. This is the luminal space, and this cell, this is an endothelial cell, because it's touching this mesengium and this luminal space. And this is that famous podocyte that is sitting on top of the capillary wall. So yeah, I mean, being in a pathology department, I learned a bit about it, reading the slides and understanding about the cells, and I use those knowledge when I develop AI algorithm. So at the heart of our tool, we have this tool called distributed human AI loop system. So imagine they're distributed because it's available via cloud. So imagine that we have multiple centers who are generating data, and data is coming from different part of the world, and they are scanning the images, which can be an individual scanning in the center or a centralized server, and it gets to a queue in our server, which is called athena.ccr.buffalo.edu, where we have this tool located. And once this goes to a queue, it goes to a CNN processing algorithm, and it shows the structural segmentations that pathologists would be interested. But as you know, that if I bring my tool today, and it is happening in HubMap in a recent discussion, that some of the data that they shared that did not work with our tool. But the whole idea here is that if the data does not work, our tool offers a way to correct the segmentation boundary. It allows you to see the segmentation either on this online viewer in the local viewer, where the reviewers can review this and they can provide this corrected data and provide that to the network for retraining a new set of data, okay, where the data, where the algorithm may be suboptimal, and then algorithm can be tuned toward analyzing this data, because at the end of the day, just solving a segmentation problem, and that may not be the bigger interest for the scientific community. We may want to know, how really diabetes is progressing. So with minimal effort, if you can segment the structures and get a large data structure, so segmentation, that would be our goal, to understand the feature diversity. So this is available. It's a content. It's a Docker image. We developed this tool. It can be developed. It can be brought to any server system and run from there. And that's what we have. So using HALE, we have made two important contributions in the area of diabetic nephropathy and chronic kidney diseases. So we developed a classification system to computationally classify target pathological staging of diabetic nephropathy. And this seminal work in the literature involves computational HALE segmentation. So human AI loop, what I discussed, I call that HALE, human AI, and L for loop. So HALE, computational HALE segmentation of glomeruli of whole slide images. We extract biologically relevant features from each glomeruli, and then use a recurrent classifier in classifying diabetic nephropathy. And then we also used HALE to, in a recent publication, we also used HALE to computationally detect and segment something called interstitial fibrosis and tubular atrophy, which we possibly, and glomerulosclerosis, which are chronic lesion in chronic kidney disease in general. And we would call interstitial fibrosis and tubular atrophy IFTA moving forward in this talk. Important aspect in this work is about application of the tool and extensive performance analysis of the tool. So our data came from multiple continents, multiple institutes, and multiple annotators were used as ground truth for reproducibility. So let's talk about this computational DNA classification work. So in brief, this work was about, as I discussed, HALE segmentation of the glomerulus, detection of glomerular subcompartment, glomerular feature calculation, and recurrent glomerular classification. And I'll discuss what that's mean. So this is a biopsy. So first stage is that, we use our HALE tool to segment all the glomerulus. Okay, that's what the pathologists do when they try to classify this tool. And the whole purpose in this work was to develop a digital assistant for the doctor for developing classification. And that's the first work, the very first work that we did. But once we segmented, right? Once we segmented each glomerulus, so this is a segmented glomerulus, this is another segmented glomerulus, we did some subcompartmentalization. So this is a subcompartmentalization structure of this glomerulus. Here, blue is the nuclei, orange is this red color is this pink region, this is the mesenterium, and then you have the luminal space, right? That is shown in green. Once those are done, we extracted biologically relevant features, and I will explain later part of the talk why we are actually instead of asking the computer to find out any pattern, we are generating we are quantifying biologically relevant features from each glomerulus. And those features could be like how big is the glomerulus, how many nuclei are there, how dense they are, how far are they from the boundary, how they are engulfed by other compartments, how different compartments are talking with each other, and all these things. I think segmentation of glomerulus is difficult, but once that's segmented, getting all these structures is a simple textbook study. That's what I even teach in our image analysis course. And once we have those, so we use something called recurrent neural network, or RNN, for diabetic nephropathy classification. And you are mostly familiar with RNN in your day-to-day life. So for example, if those who are using Gmail, where you may have noticed that when you write something, you get automatic suggestion for next part of the sentence. And this suggestion updates with more you write. So RNN updates its output with current decision. DN class level in a biopsy is assigned similarly during manual assessment. So what doctors do, they see a glomerulus and determines a level. They move to the next glomerulus. Either keeps the previous level, previous classified class or information that learned from the previous glomerulus, or they update their information. And that way they go through the whole biopsy, and then they finally come to a stage information, right? This is my, and then they say, okay, this is class stage 2 or stage 3 or whatever. So that's the same way we give our glomerulus in a series to this network, to this neural network. And Neural networks is the first glomerulus, determine a level, then the next glomerulus come, and it uses something called long short-term memory. So it either remembers whatever is there in from the previous one, or it updates based on the new information, and it goes to a series and it finally gives a level. Only difference from Tarbert classification stage is we give a continuous number than a discrete number that Tarbert provides. Then we compare the performance of what tool with doctors. And as you know that, any neural network algorithm, any machine learning algorithm, any maximum likelihood estimation algorithm, or any data fitting problem, that works best when you have infinite amount of data. Okay, I mean, as long as you tune the data, you tune algorithm properly, you optimize it, you have infinite amount of data, your error is minimum, as long as you are getting to that optimal point. But we had only 54 biopsies, and that is the sole reason why I used this handcrafted feature and used RNN. And what we did using this 54 biopsy cases, we had one pathologist was the most was used as the ground truth here, and then we had two other pathologists who we asked to label those whole slide images, and we obtained their classification same with respect to the ground truth, these two other pathologists, and their Cohen's-Kappa agreement between their classification and what's there in the ground truth that's shown here. And then computer is somewhere here. So in using computer, we are somewhere in the comparable with doctor. And what we did, since we have 54 cases in this study that we published, we also had each case has multiple sections. So we used each multiple sections as individual case, just pretended that. And we wanted to see whether the performance improves or not. And the kappa improved here with minimal error variation, and that told us that if you have multiple sections, that kind of normalizes the noise. And sometimes during the slide reading, doctors use multiple slides. So that being said, I just summarize this part of the talk. So we developed a computational DNA classification tool, and I see this nothing but a digital assistant for doctors. We used handcrafted biological feature priors, and that is very important because that helps us to understand feature diversity, and in small data context using RNN, that's very beneficial. Performance here may be limited by the ground truth. One can argue that we are actually hitting the ceiling of the doctor that we used as a ground truth to train our algorithm, and that is correct, and there is no way around in this kind of work unless we are using some sort of immunofluorescence level. We don't use tubular features, and we'll talk about that, how they are useful. We want to study in larger cohort and use image pixels as features, but that would only be used when that can only be used when we have lots of data available. So next I will go to this IFTA quantification. And regarding IFTA, one thing one should know that, I'm giving you a brief introduction of IFTA and glomerulosclerosis. So this is a crop from a chronic kidney disease, and these are the two crops, and you can see that when tubules, they are healthy, they kind of touch with each other, they don't there is not gap. But when tubules are atrophy, this whole kidney system, it's a beautiful machine, and one part start not functioning, the other part stop functioning. And what happened that in this tubule, slowly they get atrophied. So these are the atrophied tubule, and once they get atrophied, they get shrink, and there are collagenous things form in between the tubules, and that's called interstitial fibrosis. And also we have during that process, glomerulus get sclerosed, and these are the sclerotic glomeruli. This is a good glomeruli. So this happens hand in hand, and in this work we try to quantify IFTA. So in regular in doctors domain, when they look for IFTA, they visually estimate IFTA. They go through a whole slide. They don't even draw a boundary. Doctors, what they do, they give a number. They say, okay, 50% of this slide has IFTA, 25% of this slide is IFTA. And this is used as for chronic kidney disease assessment, including in diabetic nephropathy. Those who are in transplant, they know that how much this information is important, how much that I know how, like percent IFTA and number of sclerotic glomeruli in a biopsy. So what we did, we we developed a tool again to segment IFTA using this HALE classification system I talk about, and try to see, I mean, how good we perform with respect to doctors. So here are some results using different biopsies which are from the test data. So this is from transplant biopsy, green color here that shows the interstitial fibrosis and tubular atrophy. In our work, we did not separate them because they go hand in hand. Yellow color shows good glomerulus, red shows sclerotic glomerulus. It's not enough visible. And this B, C, and D, they are coming from native biopsies with DN. This is a disease donor biopsy, and this is a case which the algorithm was not trained on. So so we analyze the IFTA segmentation performance and the detection performance of the normal and sclerotic glomerulus. So here we compared the performance between the pathologist and the computer, and for this we computed intra-class something called intra-class correlation coefficient, or ICC, of 4 renal pathologists. For visually scoring IFTA means they scored the IFTA based on biopsies, and then we asked them to annotate IFTA in their slides. Then we also look for number of glomerulus that what they are counting, number of sclerotic glomerulus, and percentage glomerulosclerosis. And we tried to find out the interclass ICC measure variation, okay, when 4 pathologists were alone, and then when we included the computer, how this variation changes, and we found they are kind of similar in all those cases. So that tells us that we perform as comparable as doctors. So now let's look at that if we do pixel level comparisons, because we are getting the segmented boundary of IFTA, right? So we wanted to see the pixel level comparison between the computational output and renal pathology output. So we have 4 renal pathologists and computer, and we tried to find out that, you know, how comparable we are with doctors, and we found that, you know, we are quite comparable. The kappa is not that great, but pathologists, they don't agree with each other or either, I mean, and tremendously, and they are all this range. That's what we noticed. Next thing we wanted to see that how correlated this quantification of visually measured if the pathologist annotated if the, or computer provided if the, like, you know, C if the is computer provided if the quantified if the GS CNN is computer quantified glomerulosclerosis. And we wanted to see, I mean, because if then glomerulosclerosis, they are a measure of chronic disease, right? So we wanted to see how these numbers they correlate with the EGFR measure, because EGFR you know, is a measurement of, you know, this is a biometric, it's a physical measurement with the biopsy. So we want to see the correlation which is EGFR during the biopsy, 1 year after the biopsy and 2 years after the biopsy, and we found that, you know, they're quite comparable. R square is not much for all of them, but doctors and the computers, they are quite comparable, except for this one where, you know, the significance is not there. And that tells us that, okay, we have a tool now which currently works with the current state of the art. And then we did another study, and I would be very quick with this study for the sake of time so that I can cover the other things. So the basic thing basically we wanted to test, because HALE is a distributed system now, it is available via cloud. So doctors, now it provides us enormous opportunity to work with multiple institutes. So we use data from multiple institutes where doctors, they use this online tool to annotate, and we had 3 institution data, and we asked the question that if we train the algorithm with individual institute data, how we perform, versus if we mix the data from multiple institute, and whether the diversity matters. And we found that when we have a diverse data set, the performance of IFTA classification that improves, and that is shown in these 2 plots. Basically, this is the ROC curve for individual institute data, and this one is the ROC curve for combined data which are balanced with the individual institute data. We also compared the performance of our tool with the KPMP, Kidney Precision Medicine Project data, with 26 KPMP data, which are all test sets, where we the KPMP, there are 3 KPMP pathologists who quantified if the they provided the percentage if the in the biopsies, not they did not annotate, but they provided numbers, and we try to compare the correlation between if the percentage quantified by our algorithm and KPMP, and we got significant correlation with and comparable correlation with pathologists. And our tool provides 2 kind of outcome. One is like a map like this, in like the way it's shown in the green color, and the other is like in a heat map as shown here. Okay, heat map as shown here. So because heat map is important, because if the boundary is something, those who are in pathology, they know that sometimes doctors don't agree with the boundary, so sometimes if boundary definition might help. So this is all about using the traditional definition of IFTA. So question is, can we go beyond traditional definition of IFTA? And this is what we are investigating right now, because IFTA is something is nothing but pathologist detected boundary, right? But we are able to use computer algorithm now to segment all possible structures. For example, all the tubules, all the glomerulus, all the sclerotic glomerulus. We are not showing vessels, but yeah, we can segment the vessels, arteries and arterioles as well. So can we come up with a new definition of IFTA? For example, a more statistical measure of IFTA, because remember that we are able to extract features from each of these compartments, like how big they are, their texture, and other information. So imagine that if all these things are segmented by computer, and we can generate a data matrix for all these compartments with all their feature information, can we study those features like high dimensional data in lower dimensional space to understand about the data and about the chronicity? We have not done extensive work on that. I am hoping to do that as the next life, means my next phase of my career, those interesting studies, and study those features with respect to age, sex, ethnicity, do more work like science-based work using these tools that we developed so far. There is a preliminary work. So we took 29 transplant surveillance biopsy cases, and each dot here is the extracted glomerulus here, and from each glomeruli we have those handcrafted features I talked about, and this is a 2-dimensional representation of this data. This is called UMAP, but it's a nonlinear version of the principal component analysis. I mean, but each dot is a glomerulus here based on those features, and you can see we are getting unsupervised clustering, and we mapped this data based on the corresponding biopsies progression, which we divided in a binary format, that EGFR decline rate less than 5 per year versus EGFR decline rate greater than 5 per year. And I forgot to put this is Brandon Ginley who did all this work. He's a postdoc in my lab, so I forgot to put his affiliation. But as you can see, this red color, this is predominantly in this 3rd cluster, and we are kind of curious. This is a very preliminary work. We got an award for this poster in ASN last year by Renal Pathology Society. So all these different features, whatever like 315 features, some of the features like popped up, we kind of ranked them based on their statistical measure, and those features violin plot are shown with respect to clusters. So these are the 5 cluster and the feature expression level. And you can see that this cluster 3, which I said has some sort of a progression attached to that. There, all these features are coming mean interluminal distance or mean internuclear distance. So we can say that glomeruli from the poorly prognostic cluster have significant higher distance between luminal object, capillary loss and mesangial expansion, or higher internuclear distance, mesangial expression and nuclear loss. Things like that. So so that is the thing that we have developed, and this result is extremely encouraging, because it tells us that whatever dream I have is true, that if we can generate this big data from the large renal biopsies, pixels are already big data, but if we can kind of tune them based on the structural segmentation and get the features out, we should be able to do more intense and in-depth studies, possibly in my next slide. So so summary of computational IFTA quantification. So we developed this tool for IFTA segmentation and quantification. Then we found that our tool is as comparable as the state of the art. We tested in various data sets, in multi-compartment, multi-continental data sets from different institute with different annotator as well as with KPMP. And we have shown some promise that this large-scale feature analysis might show us a path to understand the data more and to mine the data more. So that being said, let's look at the transfer learning. Transfer learning in machine learning is something you train a model in something like a kid, right? When that kid is learning one thing and you tell them cat versus dog, slowly they start learning based on the environmental feedback and they learn about something else and they would slowly be able to differentiate giraffe versus tiger, right? So, so all these models that we have, we ask the question, can we use this model and transfer it to segment other structures? And as a first step, we try to segment arteries and arterioles. So we also have a model. So these are the whole slide image, crop from whole slide images. This B, panel B, these are coming from whole slide images of HNE, PAS, trichome and Jones, of thrombotic microangiopathy and vasculitis. Where my good collaborator and friend Jan Baker, he provided us this training data set by marking all the glomeruli, arterioles and arteries, and we transferred learned with our current model to segment the structures, and these are the 58 renal WSI data. We tested this model in GTX data in various other organs, for example, pancreas, pituitary, liver, tibial artery, nerve, and lung, and we showed this segmentation. So this blue color is arterioles, green is glomerulus, and yellow is arteries. And we also tried to test the performance, quantitative performance of our tool using MCC measure. MCC is Matthews correlation coefficient. Basically, we tried to see, whether this segmentation in multi-class context they match with the ground truth in the pixel level. -1 means complete disagreement, +1 means complete agreement. And by the way, we are using different performance measure. That could be one drawback of our work. We should use same performance measure. It's just these are different papers, different students work on this. But what interesting thing we noticed here, these are the MCC of the segmentation for glomerulus, arterial class, and artery class, where the network was started from different initial point. The network was started based on a trained model, which was trained for glomerular segmentation. That is this one. The network was started from random initialization. And then network was started based on GTX dataset classification, and network was started based on ImageNet data. ImageNet data is developed by computer scientists for deep neural network model classification, and ImageNet is based on natural images like bird, tree, dog. And we wanted to see if we train and start the network from these different models, then how they perform in segmenting this. And it quite interestingly, and I still cannot answer this, why this is happening. The network that started from glomeruli model that performs like this much, the other two initialization did not work as great, but ImageNet model always worked better, as comparable as glomerular. Sometimes it outperformed the glomerular initialization model. The way I explain that, this natural database image dataset possibly has so much variation in pixel level available that it's helping us to generalize the structural segmentation. Ideally, I would imagine that a model that is seeing more variation in pathology context should work better. But then this is something we have done. This tool is available, and what is the big picture of this work? I mean, if we can segment arteries and arterioles in places like HubMap, Human Biomolecular Project Consortium, where all data from all the body is getting generated, one significant work would be, I mean, we know that arteries and arterioles, they are like, highways, interstate highways, or, intrastate highways, and they connect the whole body. So one interesting aspect would be, if we have data available from the whole body, can we study the image features, again, in that feature engineering context, and see how similar different body organs are with respect to changes in arteries and arterioles. So that's another tool that I want to study, hopefully in my next life. So that being said, I'm very excited that it was my dream to count all the nuclei, and as you know that count all the cells, as you know, in any biology lab, when we enter, and that was the pitch for my And that used to be my pitch long ago, because when I started working in a biomedical lab, I found that it's a very common problem, like counting number of slits in a cell plate, in anywhere in an image. How many cells are there? This is a very fundamental problem, and I worked on that throughout my life. And it was my dream to count cells in a whole image context, and in kidney context, the opportunity was that how can we count all the podocytes, and we know about Wiggins method, which was published in 2014, thin thick section method in JSN, and we just this paper we got accepted. It will come in the November issue, and we got this in the cover of JSN. And my student, her picture will come later, Darshana Gavind. She did this amazing work, and also it makes me proud, because because women in a STEM area and AI especially, I mean, it's we still don't see too many, and she led this work, and it's multi-institutional, diverse data work, and developed an algorithm to count all the nuclei, all the cells, podocytes in a whole slide image context, and developed extensive performance analysis. We also have an online tool available, although they are more trained using our training data, and I'm very interested to see how it works with other institute data, and we have a long way to go. But that being said, I mean, why podocytes are important, right? I talked about podocytes, and whoever is in nephrology, and this is a nephrology and hypertension different hypertension division, so I don't have to introduce what podocyte is. Everyone possibly know it. So important. And so I'm just in diabetic kidney disease context, that that in early disease, podocyte morphological changes happen, and rate of podocyte loss predicts disease severity. So question is, can we use this podocyte counting tool for early diagnosis and track progression? And we know that, when pathologists count podocytes, they depending on the number of podocytes count, one can tell whether a patient is diseased or healthy. And loss of podocytes are shown throughout the progression of DKD. Like initially, you have 20% loss when mesangial expansion happen, and slowly it gets lost with the disease progression. And few other things about podocyte. Few other things about the podocyte is, if we can count all the podocytes, podocyte increases in size, although the tool that I'm going to show, that it's about podocyte nuclei identification, and podocyte size increases, which is shown to be correlated with progression, and then podocyte shape changes. Okay, podocyte shape changes due to mechanical forces. So Darshana, she did this work. She's not a PhD student anymore, so I need to change that. She is working proudly in Johnson and Johnson. They hired her to lead their development in AI, and she is getting more broader exposure there now, working with other data types. But what we developed, basically, we developed an extensive data set of matched PAS whole slide image. So we count all the podocyte from PAS section images, the histologically section images. That's what I did not mention, which is important, because those are the staining that are used in the clinic. And for the training with our collaborator, we generated paired training data set with same slide stained with IHC or IF to mark podocyte, either with P57 or WT1 marker. And the same slide was stained with PAS. And we also considered the case where the slide was stained with PAS first and then with IHC, and vice versa. And we found that, staining with PAS first makes more sense, because when you stain first with IHC or IF, the same slide, and then you stain with PAS, then some properties of the slides change. But we use this training data set to train the algorithm where podocytes are in this histology image, and we use two different type of algorithms, semantic segmentation as well as generating adversarial network. And using those trained model, then we take any whole slide image, PAS 10 histologically stained image, we run tail segmentation algorithm to extract glomerulus, and then we find out where the podocytes are using our model. So this is a very brief description about what happens. So we have a PAS 10 image, we have native immunofluorescence image that tells you where the podocytes are. And then we generate this in silico IF image, which we generate from this PAS image to show where the podocytes are. And we also tested with pathologists. So we asked the pathologists to mark podocytes. As you know, that marking podocyte is it's difficult for pathologists. They can mark podocyte in the boundary, but sometimes they miss whatever is in the inside, because this is a 2D card of the 3D system. But as you can see that, this is the native IF image showing the ground truth, and pathologists, they missed most of the inner podocytes here. But our in silico IF image, in our training, based because of our training, that to detect these podocytes in this data. These are some examples, and you can read the paper. It's already available online, the unedited version. But these are the ground truth podocytes, and these are the detected podocytes using two different algorithms. I think here blue and yellow shows podocyte detected using two methods, and orange shows podocyte detected using both methods. And this is the data from human biopsy. This is from human autopsy for two different marker types. Right side is the ground truth, left side is the detection. Another important question that we ask, that whether the tool can be translated in clinic. And when someone is reading slide, one important question they ask, how many glomerulus or how many podocytes we need to reliably say that our algorithm is working. We had the human data we used here. Since we had a different data sets, human data we used here has 34 cases. So whatever I'm showing here can be assumed as a thought experiment that we did to answer that question. And I need huge amount of data to develop a really a tool. But the important fact is that what we did, we had 34 whole slide images where we detected the podocyte using our algorithm, and we found the average the dice coefficient per WSI. Dice coefficient is like how good we are performing. One means that it's almost like an accuracy. One means we are very accurate in detecting podocyte. And we compared that with respect to number of podocyte nuclear count per glomerulus. And what we see that there is no, correlation. Okay, there is no correlation. That's what we found. But when we tried to compare the same metric with respect to number of glomerular profile in a whole slide image, we found some mild correlation. Although again, these are 34 cases. Okay, and we had to use some we have to use some threefold cross validation study to get this data done. And each dot is a whole slide image here, by the way. And what we found, we have a mild correlation with some people when it just tells that theoretically it is possible to set a desired performance with statistical significance with respect to the total number of WSI present in a whole slide image. Because in order to translate this clinic, first thing we have to say, look, how reliably this can be used and how reliable it is with respect to the glomerulus. And I think we have to do a detailed study in that direction, but that would be the first study or first specific aims for a clinical trial study using this tool. So an automated tool we have developed for podocyte detection from renal whole slide images. We did performance analysis, and it suggests that total number of glomerular profiles in determines podocyte detection performance. And we mine podocyte image features can be correlated with the omics features, which is what we are working on right now. But that's another thing I want to explore next. That now we can reliably get all the podocyte features. Maybe we can go to KPMP data and look at all the single cell transcriptomics data and the podocyte data, so we can get the cell level data in spatial context, morphometric context. We can get the single cell data from the genomic context, and we can compare this to data sets to mine new information. Regarding the data correlations, we have done some work, and this is the work that we have done where we try to see if we can fuse the data from image space and omics space. And for that, we have this unique data set available from Seoul National University. Excuse me for that. So we work with Dr. Sohn Sok Han. He's our collaborator. We have this unique data set of 56 DN patients available with PAS10 biopsy whole slide images. And we have urinary proteomics data measurement available for 2038 proteins during the time of biopsy for all these patients with their outcome. EGFR measurement 1 year or 2 year after time of biopsy, as well as 2-year ESRD outcome measurement. And we were very excited with this data. We wanted to ask, like, how do the image features affect the end progression? Because we work with images. If that is the case, what's the role of this urinary proteomics data, and if the image and molecular features are correlated? So we first looked at the urinary proteomics data. Here, each dot is a patient, and whatever urinary proteomics data available, we just put that in 2 dimension in a lower dimension space, where red means the people who did not go to ESRD, and blue means who went to ESRD, and we found a difference. So if we found a difference, that means urinary proteomics data can solve the problem of detection, right? But again, one fact is that these patients, they are coming from mostly aggressive disease. We asked the question, like, what are the significant proteins that are differentiating these 2 clusters? And we found that those proteins, they are mostly coming from they are in multiple compartments, that mostly coming from tubules. And we went back to single cell data study, and we mapped those 45 proteins in single cell data from 3 healthy patients and 3 diabetic patient, and this data is coming from PNAS, this publication. So we mapped this protein data in their data they published, and we found the basically here, this is for the control data set, this is for the disease data set, and we found that our intuition was correct. Those proteins, they are mostly coming from the tubular compartment, and as you can see, all we developed in diabetic nephropathy context in the previous part of the work, they are from glomerular. So question is, maybe tubules has some role, and it's written in the textbook, but I wanted to verify that. And we exploited this multi-compartment segmentation, that tool that we have. So we have multiple tubules available now. We have glomerulars, we have sturdy glomerulars, we have vessel segmented, and like before, from each tubule we did bit of a internal segmentation, each nuclei, lumen, and as I said, once the tubules are segmented, that is the hardest task. Segmenting this internal structure is not difficult, but what we did, we developed handcrafted features like glomerulars from each tubule as well. And we first tested the performance of the urinary proteomics data, and this is a confusion matrix, which is the ground truth versus predicted, to find out a number. And 0 means no progression, 1 means progression, and we found that urinary proteomics data is very good. They are going to a 0.91 Cohen's kappa to predict the outcome. Then we used our RNN model to see what the glomerular image features and tubular image features offer, and we found that glomerular image features are not at all predictive. They have like a poor Cohen's kappa 0.14, but the tubular image features are providing 0.49 Cohen's kappa. So that means tubular image features, they have actually some information or more information than glomerular image features. Now, question is, I mean, the urinary proteomics features are already much better in predicting progression. So I think one way we are trying to see, that, I mean, traditionally the classification system, they use the glomerular image feature. They don't use the tubular image feature. But if the urinary proteomics data, they are very non-invasive. I mean, can we combine all this? Can we learn from this data to come up with a new classification system? We are working on this, and I will just show you some results here. I mean, we transferred this urinary proteomics data in pathways. So we have 21 pathways that we got. We have their pathway score. We use a simple scoring system using engineered pathway analysis. I'll not go into the detail, but we have the image features, and we try to look at the correlation of the image features with the pathway, and we just looked at where the highest correlation is happening. And one of the correlation is this one, which was a correlation between mean area of object with PAS positive distance transfer value versus coagulation pathway. Although the image feature I'm talking about, again, mean area of object with PAS positive distance transfer value, no one understands this, right? So ideally, it's better that we project that in the image space. So when we see that in the image space, so this is the patient with ESRD, this glomerulars, versus patient with no ESRD, and we see for that glomerulars, this particular feature is showing some distinct difference. And then the pathway score, when we see from non-ESRD versus ESRD patient, they have some significant difference. And and that tells that we can generate new hypotheses. So we got several candidates along this direction with image features and pathways and the proteins, and I believe that would need some group science work with a molecular biologist to get to the detail of what we are looking here. I mean, my work stops at the feature analysis, doing large scale data analysis, getting this feature map, but then we would possibly need molecular biologists who would be a co-PI or co-invest to understand these features, or maybe study some of these hypotheses that we are getting. So that being said, as I said that tubular image features are important, they're predictive of outcome. Renal image features are correlated with important DNA pathways, and we got new pixel level map with respect to molecular pathways, and it required some biological validation. That being said, I wanted to thank my collaborator Kwang Eugene. He's a significant collaborator. We work with him, Avi Rosen from Johns Hopkins, Dr. Han from Seoul National University, Jan Becker, also work with John Tomajaski in University at Buffalo. These three students are at the center of our work, my entire lab, and we are mostly funded by NIH and NDDK. I recently got this HubMap grant as a co-investigator for a year, but I hope that I would be able to make a compelling case to get more funding from there. And that's all. I think I'll be happy to take any question that you may have.
Thanks, Pinaki. That was a great presentation. Very interesting, and congrats on all this high impact JSON publications you've had. So being part of the ASN family, I know that this has had high impact in the nephrology community. As people get ready to ask you some questions, one quick question: have you noticed anything between the difference between podocytes and tubular structures in your prediction of outcomes? I mean, obviously, classically in nephrology, we've been focused on the health of the tubule and the atrophy of the tubule as a predictor factor of progression. But I mean, this machine learning project that you have on podocytes and measuring numbers of podocytes may change things. Do you have any data?
We have not studied that, but we have definitely tools, and we have cohort available in diabetic nephropathy context. We can definitely look at the feature correlation, and that would be interesting. Yes.
Thanks, Pinaki. Anyone with other questions?
Yeah, I think the podocyte and the progression definitely depends on these tubular changes, the way we understand the system biology. So,
Pinaki, this is a really cool presentation. What was interesting, one of the interesting things about the podocyte project is that you were able to take immunofluorescence and then integrate it, I think, into the PAS analysis. Obviously, when we stain biopsies, we do immunofluorescence and then other stains like silver stain, et cetera. Do you kind of see it that you could take the findings of those other stains and again, train the PAS analysis to find the same features? Or do you see more that there would be separate analyses of different stains that would be their own data set?
I think, Josh, the way we did the work in this JSN work, we can in fact use all the stains and we can integrate that. I mean, it is so far our learning, and this is what I learned. This is a extremely the data set can be extremely heterogeneous, but it is our learning that once you have this immunofluorescence or immunohistochemistry data, we do some sort of a pre-processing to get another level of map where we show from the ground truth where the podocytes are, and we use this secondary data to train the model. We don't use directly immunofluorescence and immunohistochemistry data because that did not give us enough performance. So we can take any sort of data which has this podocyte ground truth, we can convert that to their common data type, and then we can train our model. I think the interesting thing what I noticed, and which the community should know, that in order to train this model, you need a PAS data, right, from the same section. And ideally, we found that if we stain with PAS first, and then if we do immunofluorescence or immunohistochemistry, ideally immunohistochemistry, you cannot do immunofluorescence. It's much better because you don't change the PAS quality, But if you do the other way, then it changes the PAS quality, and then machine starts learning those noise. It's bit so that's the major learning that we did.
This is Larry Chen. I have two questions. Wonderful talk. So
Thank you.
what spatial technology are you using? MIBI, Mectra, or IHC, that's number one. And number two, your pixel technology, can you incorporate this for the imaging data with the basic, for instance, for MRI, you have the biophysical data, T1, T2, those into this pathology pixel data?
I did not understand the first question. The first question is what technology you are using, right, for this kind of work, right?
What spatial technology, because you have MIBI, the Vectra, and IHC for the spatial technology in your HAL program. So,
I see. So right now the HAL works with PAS and HNE data. That's what our core lab develops. But for the immunohistochemistry, it's a standard, immunohistochemistry or immunofluorescence in our lab. We recently tried to work with the codec or Kodak system data, which provides, multiple fluorescence data. We are not very successful with that, because when you use multiple fluorescence to train this kind of model to get multiple structures, we found lots of bleed through in different channels. So that part we are still optimizing, and it was an enormous amount of cost for us to generate that data. So we are going slow with that. And your second question is, yes, MRI and CT data, yes, that definitely can be integrated. And the question that I'm trying to resolve these days, and I'm hoping that if I can get some data set. So you see, when someone is taking a biopsy, right? I mean, that is my question, a fundamental question. And I think that's one thing that in HubMap, they mentioned they want to be interested in. That if someone's kidney, if you take a biopsy, right, you don't know where it is coming from, right? You possibly know a little bit, but then if you have the CT data, how can you map this 2D data, whatever 2D segmentation we are doing in the 3D space? So I'm very interested to solve that problem if we have matching data set available, and it would be intriguing to work on that.
Thank you. Very good.
Thank you so much, Pinaki. Any other questions? Again, Dr. Sarder will be meeting with many of the faculties here in Renal and other departments. So, Pinaki, thank you so much for this presentation, and we look forward to speaking with you and meeting with you one-on-one.
Thank you.
Thank you. And if you have any questions during the day, don't hesitate to email Rachel.
Sure. Please.
Okay.
Yeah.
Thank you.
Thank you.
Thanks, Pinaki. Bye-bye.